We are the paradoxical ape. Bipedal, naked, large-brained, long the master of fire, tools, and language, but still trying to understand ourselves. Aware that death is inevitable, yet filled with optimism. We grow up slowly. We hand down knowledge. We empathize and deceive. We shape the future from our shared understanding of the past. CARTA brings together experts from diverse disciplines to exchange insights on who we are and how we got here. An exploration made possible by the generosity of humans like you.
Hello everybody, thank you for the introduction. I also would like to thank the CARTA leadership for inviting me to this symposium, and I thank everybody that is watching for coming. Today I'm going to talk to you about LINE retrotransposons. But before I get started, I really want you to know the take-home message that I want you to know for today. First one is that the neighboring cells in your body are not identical. The second one is that humans restrict how different those neighboring cells can be a little bit more than apes or non-human apes. So with those in the back of our minds, now let's start with the exciting news. The exciting news is that what we learned in school about our own DNA is not completely accurate. The concept that all the cells in our bodies have the exact same distinct DNA, and that of course makes us very unique, meaning that me and my best friend, we're different, but all the cells in my body have the same DNA, and all the cells in the body of my best friend have their own DNA. That concept has been challenged due to new DNA sequencing technology and models, both in vitro and in vivo, that can test this concept. The idea now is that we are mosaics. And that means the cells in our body are not genetically identical. That's especially true for a brain. But the good news is that you are still unique in your own mosaicism. So one of the culprits for those differences in that mosaicism in our genome is our retrotransposons. So retrotransposons are a class of mobile elements or jumping genes that are able to copy themselves, and they're present in our genome, and they can insert elsewhere in the genome. Those elements were initially discovered, the transposon elements were discovered by Barbara McClintock. She got a Nobel Prize for that discovery, and she was not able to explain the inheritance of those kernels in maize that were different. She could not, had different colors. She could not explain by using typical Mendelian laws. So she went deeper into the genetics of what was going on, and she identified mobile elements, those DNA elements that were moving around and were causing that diversity on top of what would have been expected by typical Mendelian laws. Specifically the ones that I am going to talk to you about today, those are called LINE1. And LINE1 stands for long interspersed nuclear elements. And they are the only autonomous mobile elements that are active in humans. To have an idea of how many are there, they comprise about 20% of our genome. In comparison to coding sequences, that are the sequences that make genes, those are about 2%. So they are very abundant in mammalian genomes, and specifically human and non-human primates. So what would be the consequences of these elements moving around our genomes? Imagine that you have a sequence in your DNA being copied and inserted elsewhere. So that new insertion can affect chromosome integrity, cause diversity, and can also generate changes in behavior and potentially diseases. So the germline insertions can cause those structural variants, deletions, sequence insertions within a human population. So they happen early during development, either in the egg or in the sperm, and they can be passed along to generations and impact population. In those can potentially cause diseases, and the first evidence of disease caused by L1 insertion, LINE1 insertion, was hemophilia A, resulting from a new insertion of a line sequence. This was described decades ago by Kazazian. To date, over 120 human diseases are associated with LINE1 events. So for a long time, it was thought that all the mobility, the LINE1 mobility, was happening at the germline time of development. Nowadays, we know that LINE1 insertion can also take place during embryonic development and even in adulthood. So during development, given the copy and paste nature of the retrotransposon processes, LINE1-driven insertions can accumulate. So as I mentioned before, they can happen at the germline levels, which can have populational consequences. But they can also happen what we call somatic, which means not at the germline, but later on during development. They can also happen at the embryonic development, and they can also happen at the adulthood in organs. And I'm giving you an example of the brain here. So how do we study line mobility during development? So especially human development. So first, you choose a cell line that represents human development. And luckily for us, research from Shiina Yamanaka in 2008 made it possible for researchers to study pluripotent stem cells, which are similar to the earlier developmental stages, where those cells are now possible to become different any other cell in the body. So those pluripotent stem cells are able to become neuroprogenitors and neurons, for instance, but also other cells in the body. So we can use these lines in the research laboratory setting to study human development aspects. And we can look at, indicated here by those red arrows, you can look at line retrotransposition in these different stages. Another way is, or an additional way to study line mobility, is to choose a system that will allow for monitoring line mobility in real time. So we took advantage of a genetically engineered LINE1 element, and that element can allow us to see live mobility happening. How do we do that? It's an engineered element by John Moran that will show GFP, or enhanced green fluorescent protein, glow after one round of retrotransposition. So what that looks like is we see those green cells in the dish of tissue culture. And when we have a green cell, we know that we were able to see new insertion. So with that in mind, this is work from many years ago, we set up to look at earlier stages of neuronal development. So those are the baby neurons, so the neurons that the progenitor cells that are going to become neurons if we coax them with specific factors. And we used induced pluripotent stem cells, so those cells that we could make from patients' skin, and we use cells from Rett syndrome, patients with Rett syndrome, which is under the umbrella of the autism, has some characteristics from autism spectrum disorders. So we took those neuroprogenitor cells highlighted here on the top, and we added the LINE1 indicator cassette to be able to see LINE1 activity in real time. And we not only detected LINE1 activity on neuroprogenitor cells here called NPCs from healthy individuals, so those are the called here wild type WT, but also we were able to see an increased activity, an increased real-time jumping or mobility of LINE1 elements on Rett syndrome patients. So that highlighted to us that line mobility was happening in human diseases at potentially earlier time during development of the brain. We went ahead and looked at potential causes for that increased mobility in the cells derived from these patients. And we know that the gene that represses, one of the genes that represses LINE1 expression called MECP2, it's a methyl CpG binding protein. It's not functional in Rett syndrome patients. So just to give you an idea of how this gene works, it occupies a portion of LINE1 element in the genome that is responsible for the control of that element. And when you don't have MCP2, here there's a yellow, showed in yellow, you will not have LINE1, or you have you don't have LINE1 repression, so LINE1 goes up. So mobility of LINE1 can be regulated in neuronal progenitor cells and could play a role on complex neurological conditions. Since then, other researchers showed involvement of LINE1 activity in other neurological conditions such as schizophrenia, for instance. Now, we wanted to shift gears a little bit and look at evidence for LINE1 mobility in evolution, and we wanted to see if we could use the current models that we had in the lab to study that. So we went even further back into the developmental process, and we used those pluripotent stem cells to look at LINE1 mobility in real time. So we took human bonobos and chimpanzees, so those are our closest living relatives, and cultured those induced pluripotent stem cells from all of those of these different species. And we looked for evidence of LINE1 retrotransposition in these stem cells from the different species. And what we noticed, we used the same indicator that we had used before. So we're looking for green cells, the GFP positive cells. And what we noticed is, yes, we do see mobility in human cells, but we do see a significant increase in the number of LINE1 positive cells in bonobos and chimpanzees, quantified here in the right. So we detected increased mobility in chimpanzees and bonobos, and associated with that increase was also a decrease, so here showed in green, of those two genes that are known repressors for LINE1. So they were shown in red, were upregulated in humans, so there was more of the repressor proteins in humans compared to chimpanzees and bonobos. So that means that LINE1 was repressed in humans in part by the increased presence of those two proteins, also shown here by those very dark bands compared to chimpanzees and bonobo lines, which had lighter bands, so less presence of the repressors. So since they didn't have the repressors present, the chances of the line 1 to be higher and jumping more was higher too. So line 1 was repressed in humans, but not chimpanzees and bonobos during embryonic development. So we asked the question, does increased line 1 mobility in non-human primates, in chimpanzees and bonobos, as we've seen previously in the cells, does it result in more line 1 insertions, more lines in the chimpanzee genome compared to the human genome? And to answer that question, we collaborated with Chris Banner at UCSD, and also Inigo Narvaeza and Ahmed Tenli that were both at in the lab at the time, and we compared the presence of abundance of the elements in the genomes for various subfamilies, different subfamilies of line 1. So the way we looked at that is we compared the older line 1 elements. So we looked at both genomes, human and chimpanzees, and we searched for older line 1 elements first, the ones that were present before the split between the species. So those are the L1PA4, line 1 PA3, and PA2. And when we look at the older elements, we didn't see a significant difference. Now, when we looked at the newer line 1 elements, which are present in the genomes of all human or chimpanzees, because they are after the split, we did see a significant difference, where line 1 from primates, non-human primates, more increased. So there were more abundance of sequence from greater representation of recent line 1 content in chimpanzee genome compared to human genome. So we hypothesized that increased line 1 insertions in chimpanzee genomes could potentially contribute to increased genetic variability in chimpanzee populations. When we looked for evidence for increased variability in chimpanzees compared to humans, we found some data for that in literature. Work from a laboratory of Evan Eichler looked at great ape genetic diversity and population history, whole genomes of 78 great apes. And what we're showing here is genetic diversity. I'm showing these images, genetic diversity measured by expected heterozygosity. What it does, it will describe the expected proportion of heterozygous genotypes under Hardy-Weinberg equilibrium. Genetic diversity is the probability that a pair of randomly selected alleles from a population is different. Let me say that in different words. So what this measurement does, the expected heterozygosity, it compares set of genes that you get from mom, set of genes that you get from dad, and asks the question, are they the same or are they different? And what is the proportion of the different that you have different mom from dad in your population. So, and that's called heterozygosity, when you have different sets of genes from mom versus dad. So if your population has a high heterozygosity, that means there is more diversity in the population. So in red, I'm highlighting the expected heterozygosity in humans and expected heterozygosity for many of non-human primates. So from that study, you can conclude that non-human primate species have increased heterozygosity, which is one way to measure genetic diversity compared to humans. So that's one of the examples. So we think that this increased diversity in non-human primate species could have been driven in part by the increased line 1 activity. So the summary and implications of our studies is that line 1 elements are more active in non-human primates than in humans themselves. We also shown that species-specific line 1 elements are more abundant in non-human primate genomes than in human genomes, especially the newer elements. And we also have some evidence in the literature showing that genomic diversity in non-human primates is increased. And we think that that could be potentially driven in part by LINE1 activity. So what are the implications of our findings? So longer brain development and cortical expansion in humans involves many successive cell divisions. If you have uncontrolled LINE1 mobility, that can be a dangerous liability to proper brain development. Now, we speculate that a more stringent LINE1 suppression during early development might have been required in the lineage that is leading to modern humans. And with that, I would like to thank you all for coming and thank Rusty Gage, Inigo Narfaiza, Armand Deli, and Chris Benner that were directly involved in this work. I would also like to thank a very bright and amazing group of women that has helped me a lot over the years for this project and associated projects. And I also would like to thank my new CSD students that are going to be taking some of the non-human primate work on, Isabel, Autumn, and John. I would also like to thank CARTA and the Comparative Anthropology series for inspiring this work. And thank you all so much.